How's it going, everybody? We're going to look at Chapter 5 today, and Chapter 5 is going to focus on eukaryotic type organisms. So I talked about a little bit of the difference between the two major cell types back in Chapter 1, and these are the prokaryotic and the eukaryotic cells. So Chapter 4, if you recall, talks about the prokaryotes, mostly bacteria. And in the eukaryotes, we walk into a more complex topic and have less time to go through as much of the broad information. But we're going to talk about eukaryotes with trying the same amount of information as we had in Chapter 4. So we'll see it doesn't go as far, but we are essentially looking at the more complex of the two cell types, and these are the eukaryotes. Start by just some visuals. These are images off the web, all real images, high-resolution microscope images mostly. These are a handful of very unique, but all members, unique cells, but all members of the eukaryotic kingdom. So you can see how much variation we have here. So as a result, we don't really get into stuff like cell shapes like we did in the prokaryotic topic. There's too many cell shapes. There's not a small handful we can narrow down. We won't talk quite as much about the generalities of appendages, for example. We'll talk about a few, but there's too many different types. So we don't go through as many fundamental things as we did in the prokaryotes simply because of the variation, and it's pretty intense. So we go over much broader aspects without as many similar generalizations that we can make. So let's start here with the definition from the textbook of a eukaryotic cell. Not the best definition, but it defines it as a cell that differs from a prokaryotic cell, going on to say having a nuclear membrane, i.e. a well-defined nucleus, and then it goes on to describe membrane-bound or subcellular organelles. And I usually refer to these just as cell organelles or cellular organelles. And then it also mentions something about cell division. If you've taken general biology, you should have studied mitosis, and may or may not have compared that to another process called binary fission, which we will talk about later in the semester, but that's a different way of dividing a cell in two. So part of the definition, not something that we're going to focus on this last part here. So what we want to focus on to start with, there's some of the major differences. As I mentioned, there's so many subtle differences, we can't look at those. We have to look at some of the underlying or overlying themes. So one of the ways, two of the ways that scientists really break up the groups, prokaryotes and eukaryotes, is that they look at the basic cell features. And there's two primary features that we can look at in a general way to loosely group these two types of cells, prokaryotes and eukaryotes. And these two primary factors are, number one, the way the DNA is surrounded by a double membrane. Let me pull up a quick figure here from our textbook. This is a general eukaryotic cell. It's designed to give a generic shape and an overview of what you'd find in it. But here we see a nucleus kind of cut open as we've done a cross-section out of this cell to show internal detail. But here you see a nucleus, and this membrane here represents kind of a special housing for the DNA, not shown here, that would fit inside this nucleus. There'd be so much of it that you wouldn't be able to see anything if they actually put the DNA in there, so they just kind of show it empty. But this would be filled with nuclear material. And this membrane separates that, and it creates a distinct boundary between the rest of the cell and the genetic material. Now, DNA is pretty important stuff. You hear about the power that DNA can have in our the way we look and the way our health, and whether we develop certain genetic diseases or ailments. So DNA is powerful stuff, and the more organized it is, the better off the cell is as a whole. And that's kind of a good way to think about it without going into too much more detail. Okay, so that's one general difference. And in the prokaryotes, they don't have this, and the DNA is just kind of free floating in the cell without a special membrane, and that creates a significant difference that carries over into other layers and other aspects of the cell. The other big difference is similar but slightly different, and that is that they contain cellular organelles. And cell organelles are a lot like the nucleus, but they are categorized separately. But some examples here include the Golgi apparatus. We'll talk about that. The mitochondria. We'll talk about that. Endoplasmic reticulum, both rough and smooth, shown here in this area. what each of these are is a special location of the cell, referred to as the organelle, but it's a special location with a function that's specific to each component, each part, and each area is somewhat walled off from the rest of the cell. And if you remember from chapter 1, I made the analogy of a single-room apartment in a prokaryote versus a multi-roomed house in a eukaryote, meaning that one is like a one-room apartment, the prokaryote, and the eukaryote's like a room, or a house with many rooms. Well, each one of these membranes that separates an organelle is kind of like the walls within a house. So, for example, we can see a membrane surrounding the Golgi apparatus shown here, and it's almost like its own room. It's like a room within a house. It's got its own larger container, but within the cell, we have even more separation. So this can operate under one set of conditions, while another component over here operates under a different set of conditions, kind of like how you might have one room in the house warmer and another room colder. And this gives the cell more dynamic control over each little area. And again, that translates into a much higher level of functionality. Okay, and this is just a real small condensed version of the prokaryotic cell from chapter 4. You can refer back to that from chapter 4, but you don't see any of that. It's all just kind of one container, and under this scenario, everything has to more or less operate under the same conditions, and that can certainly work. But this is more advanced, and it creates more dynamic control. So, looking back at the PowerPoints, then you'll want to focus on these 2 points here. Now, we'll talk more about some of the other differences. These are not the only differences, but these are the 2 primary differences. And there's a question that you'll be asked, what are the 2 primary differences? And so there they are. Another question on the review coming up is in reference to the 4 kingdoms of eukaryotes. So eukaryotes themselves are their own cell type: eukaryotic cells, eukaryotic organisms, or eukaryotes. But they themselves are all each very different as well. So we won't necessarily go into each one of these in a lot of detail, but I want you to recognize the 4 major groups of eukaryotes. So you have plants, animals, protists, which you may not be familiar with, and then the last one are the fungi, the fungus. These are the 4 kingdoms. Plants and animals, you know what those are. Protists, as it says down here, are members of a single-celled group. So it's actually a kingdom protista, protists for plural, and they are loosely categorized as being single-celled eukaryotic organisms. So every member within that group is single-celled. Now, I'll say that with a bit of disclaimer, because this group, there are some exceptions to that, actually. And when it comes to the algae are some exception, and I don't want to get into that because that's a whole other topic. But they're actually looking to break this apart and maybe create more kingdoms, and this may change in the near future in terms of how many and what they mean. For now, it's still listed this way, but recognize this actually will probably change in the near future. But that's not something you have to worry about for this class, just a FYI. Okay, so 4 kingdoms, you want to recognize those. Another thing about the eukaryotes I like to always point out is they can be both single-celled and multi-celled forms. And another way to think about this is that they can be both microscopic or invisible to the naked eye and visible to the naked eye. So when it comes to the prokaryotes, they're always single-celled. So in that group, you never see them with the naked eye unless you have a huge colony or something where millions are growing on top of each other. But individually, you don't see them. When it comes to eukaryotes, if you see it with the naked eye, such as an animal or a large plant, then it would be a eukaryote. So all eukaryotes are macroscopic and in multicellular form. But then there are also single-celled versions of eukaryotes as well. So the microscopic world is made up of both prokaryotic and eukaryotic organisms. The macroscopic world, however, really exists only with eukaryotic forms. We don't see macroscopic prokaryotes. There's no such thing. They're all single-celled. So recognize that there's a couple fundamental differences there in terms of what we see every day with the naked eye. These are all eukaryotes, but then we have a whole invisible world that consists of both types. A couple more things about eukaryotic cells. On average, they're about 10 times larger than prokaryotic cells. And this is on a cell-to-cell comparison. So not necessarily a multi-celled organism. This figure here, as I was showing earlier, gives an approximate size ratio of the eukaryotic to the prokaryotic cell, like a bacteria shown down here. There are some exceptions to that. There are some prokaryotes larger than some eukaryotes, but on average there's about a 10-to-1 size difference, and that's pretty significant. Also, when we talk about the complexity of eukaryotic cells, I use this word complex a lot, but I just wanted to mention a few examples here of what that actually means, and we'll talk about most of these. A cytoskeleton, for example, is really a whole infrastructure that is created around a eukaryotic cell to allow bigger size and allow for a more advanced internal structure. Think about it in terms of a house. If we wanted to build a large 2-3 story house, we can't just start stacking rooms on top of each other. We need a good infrastructure. We need a good foundation. We need good walls. We need good large support beams. And all these things allow for the house to get bigger and bigger and bigger. So it supports itself. And cells have similar features. And this is a whole structure called the cytoskeleton. The way cells move around and their motility is much more advanced than in a eukaryote than in a prokaryote. There's actually some pretty similar features. We'll see flagella exist in both, and the name applies to both. But in the eukaryotes, the flagella has a greater range of dynamic motion and is ultimately able to do more things. We see, again, that term membrane-bound organelles, and I probably shouldn't have repeated that, but the idea is that, once again, we see a great range of internal structures that have their own functions. And then the last one is from the definition. It was mentioned in the definition. Mitotic cell division is a specific type of cell division which involves one cell, parent cell, splitting apart to form two cells. And this is a much more complex version of a process called binary fission. And that's something that we will talk about, especially in lab, coming up in the semester. But ultimately, that's something I just mentioned as an example. I won't really talk about that at this point. Okay, real quick, we're going to pull up a document, a paint document, and do a quick comparison here. So real quick, go ahead and pause this if you want to write these down real fast, and then jot down as I go through my document, my paint document here, each of these points here. So we're looking at a figure. We'll do just a real quick, hand drawing of a prokaryotic to a eukaryotic cell. We'll check the reference and size, and then I'll point out some cell features or internal structures that are found in both cell types, and then some that are found only in eukaryotes. Okay, and then we'll move on to the rest here. So do this kind of quick. So we want to start by just using some basic cell patterns. Here I've got my little rounded edge box shape. And if we were going to draw a eukaryotic cell, we could start with a cell about this big, and with the prokaryote, a relative size, we'd want it to be about that big. That may not be a perfect 10-to-1 ratio, but it's probably close. So here's our eukaryote, here's our prokaryote. Okay. So there's our size comparison. Now, one of the other things that we want to look at really is the internal complexity. And I don't want to go into every specific example here. I just want to give you something to visualize, and I think it helps to just kind of doodle a little bit. But the idea is that inside of a eukaryotic cell, we have these separate divisions within, whereas in the prokaryote, it's all one space. And I know I've repeated myself a number of times here. But the idea is that we can start to break up the cell piece by piece. So for example, we see a nucleus here in the eukaryotic cell, and we won't see that here in the prokaryote. And the nucleus, again, is that membrane surrounding the DNA. Surrounding this, we'll see another structure here that would attach, and this is called endoplasmic reticulum. Move that real quick. Another structure usually attached to the nucleus. We'll see other structures in here then called the Golgi apparatus. Each one of these their own separate space within the cell. And then as another common example, we'll do one more here. These are not all of them, by the way, but just one more common one. we would see a structure called mitochondria. So each of these here would be an example of a structure found just in a eukaryote. So we're looking at what internal structures are found only in eukaryotes. Sorry, I'm going a little bit out of order here. Now notice I haven't really done anything to the prokaryote, and that's somewhat intentionally. Now we will add a few things in here. So let's talk about a few things that would be common to both. One of these would be genetic material, which, if you remember from chapter 4, we mentioned refers to DNA. Oops, come on. My D is not working. DNA and RNA. So these are the two types of genetic material. I was going to capitalize that. My capital D doesn't work. That's weird. Anyway, so genetic material would be found in both cell types. Now, where it's found would differ. But in a prokaryote, for example, DNA especially would just be floating around in the cell, just somewhere in the space like everything else. In a eukaryotic cell, the DNA would be specifically found in the nucleus here. Okay, so we see just a general space here versus a specific space here. Now, RNA, we won't get into that one right now. That has a little bit more of a specific type of role. Another structure, but to clarify, it would be found in both. Another structure would be a series of them called ribosomes. And ribosomes are protein synthesizers. I mentioned those in chapter 4 as well. Once again, in the prokaryotic cell, they're just floating around in here. But in a eukaryotic cell, these are actually highly organized. At least the majority of them are organized within a place called the endoplasmic reticulum. Now, there's a little bit more specific spot called the rough endoplasmic reticulum, and we'll talk about there's essentially two types of this, but ribosomes are found in this portion of the cell. So these would be structures that are found in both, and represent some of the basic building blocks of all life. Another structure, we'll do one more here, and another structure that would be found in all living things would be a cell membrane. And we'll talk about these as well. Now, this would be fairly consistent in terms that the cell membrane would be found around the outside of the cell. It's what essentially encloses the cell from the rest of the outside world. Now, sometimes there's structures in addition to that, like a cell wall, and in the eukaryotes we'll see cytoskeleton. In bacteria we saw capsules and things like that. But essentially these represent the closest boundary to what's inside the cell, and both cell types would have this here. Okay, so these are some general differences: size, internal complexity, and examples here in the middle. Just to clarify, these in the middle would be found in both types. Okay, so that answers all those questions there, and you can refer back to that later for a quick visual. All right, here's part 1 review. Go ahead and pause, take a look at this. The last one here is just a reference to that. I don't really have any specific questions. The questions are actually already on the last part there, so just refer back to those and use that as a visual. Okay, the next part gets into the specific aspects of a eukaryotic cell, and we're going to go through this list here in some in more detail and some in less in detail. We're going to start with appendages. The book refers to the next section as cell boundaries. This is the equivalent of the cell envelope in bacteria. And then we look at the organelles, and then we'll talk about the ribosomes, because they're not technically an organelle. They're a different type of structure, but we'll talk about the prokaryotic versus the eukaryotic forms. Okay, so we're going to take a look at motility appendages. And again, we're going to go through kind of the same order I went through the prokaryotes with. So starting with the outside, working towards the inside. Now again, as a real quick reminder, there's so much variation that we're going to stick to the most consistent features of a eukaryote. Otherwise, we could talk about variations in these for the whole semester. And there's really whole classes dedicated to each of the kingdoms of eukaryotes, and then even further divisions within there. So there's too much variation to get into all the details. But so, as a general example, we see a structure that is also called the flagella, found in eukaryotes, just like we saw in the prokaryotes. And just like in the prokaryotes, it is used for movement as well. So it's a tail-like structure. It extends from the membrane outward and acts in most cases as a propulsion device. These, much like the prokaryotes, respond to sensory information. So they get stimulation from, say, light, for example, or they can get stimulation from chemicals. And these will trigger movement either towards or away from some sort of environmental stressor, whether it be the light or the chemicals. Now, the one thing I do want to point out here is that there are a few differences. So the name and the function is pretty consistent between the pro and the eukaryote. Where there's differences is in the actual arrangement and the structure of the flagella. And it's said to have what's called a 9+2 arrangement. And what that means, here's a figure. What that means is that the flagella is actually made up of a series of interacting tubes that are then surrounded by their own portion of a membrane. Now, this is much more complex than the prokaryotic flagella, which really just consisted of a device to anchor it to the membrane, and then a tail-like structure that kind of hangs out from there. You know what, let me pull it up real quick. Here we see the prokaryotic flagella, and this may not be the best picture, but we have really just one big kind of structure here. It doesn't have a bunch of internal complexity, and it's just kind of attached to another simple structure, and then these rings here will rotate to move that flagella in a couple basic ways. Now, it's, still relatively complicated, but if we compare that then back to the figure we were looking at a second ago, internally this is a whole nother type of structure here. So these structures form their own little ring, and then internally are connected to these around the edge here. And the way this works is that these two middle tubes will slide past each other. And what this does, these middle tubes here, when they slide, they create a tension that will ultimately move this entire flagella one way or another. Okay, real quick, I'm going to pull up a video and show you just real quick what a flagella would look like. So this is a video off YouTube called Cytoskeleton Microtubules. I'm kind of fast forwarding here to spot 2 minutes and 27 seconds, and it's going to get into the structure of a flagella, and this is also similar in a structure we'll talk about next called the cilia. And what they're going to essentially show
one pair
is the 9+2 arrangement here. So here we have the 2 pairs of tubes in the middle, followed by 9 pairs around the outside, called the 9+2. And I'll let the video talk for itself, but this is going to act as a way for the flagella to move. So it'll show you how that works here.
Pair grows from the center of the basal body, while the other 9 pairs form the outer infrastructure of a cilia or flagellum. A gap the width of a microtubule exists between each of the outer microtubule pairs. Numerous motor proteins firmly attached to one pair of microtubules have legs that cross the gap and can walk on the adjacent microtubule pair. The force applied by the motor proteins causes the microtubules to bend.
So what's happening here is the 2 tubes in the middle will rotate past each other, and because they're still somewhat connected, this causes one side to stretch kind of like this, and the other one contorts with it. So what's happening is one side's moving and the other side's bending. So as one pulls down, the other side pulls towards it, and the other side pulls down, it pulls towards it like this. So that provides tension internally, and that allows this to move up and down.
The synchronized movement of large columns of motor proteins attached to microtubule pairs trying to walk up or down adjacent pairs creates the rowing motion of cilia and the wave-like motion of flagellum.
They're nice. I'm not sure if that really explained it fully, but the idea is that this has a much greater range of motion. And in the prokaryotes, the flagella really only does 2 things. It either moves in a straightforward kind of fashion, or it moves in a random orientation. And that's pretty much it. One's either straightforward or random. And through trial and error, microbes will eventually move in one general direction or another. but they can't necessarily turn on a dime, or they can't necessarily guide their motion very specifically. With a eukaryotic tail, they can do just that. They can move in 3 dimensions, and this tail can be quite accurate in terms of its placement. And they do come in a great variety of shapes and arrangements. So we won't get into that. But that's the idea of the eukaryotic flagella, how it's different. And to clarify here, the term that you're looking for is it's said to have 9+2 arrangement. And that's not necessarily in reference to the grouping of the tails, but in reference to the grouping of the tubes within the tail that make it move internally. Okay, back up here and just take another look at that real quick. And that's really the main thing I want you to recognize. 9+2, greater arrangement, or greater complexity, excuse me, and look back here at some of these basic similarities here. So cilia are similar to flagella in a couple ways, and that is, in one way, is that they both have the 9+2 arrangement. And if we go back here, we see cilia here and flagella here, and they both can be shown in the same diagram here, representing the 9+2 arrangement. So that is similar. In addition, they both can serve for movement, and in the cilia, these act as little oars that move an organism with like lots of little propellers. I make the analogy of a Viking boat with lots of oars on each side, and each one just provides a little bit of movement. Whereas in the flagella, we see a bigger whip-like tail, and that provides a more central mechanism of movement. Here we see organisms with 2 flagella, but one big propulsion device versus many smaller ones is really the difference in terms of how movement works. Now, another feature unique really just to the cilia is a feature or function that the cilia serve that is filter feeding, and these are called oral cilia. Now, some organisms will have cilia for both movement and feeding, and then some will have cilia for one or the other. But in general, across the spectrum, cilia can serve 2 functions, movement and filter feeding. And go back to this figure here. In this organism, this is called a paramecium. It uses the cilia mostly for movement. In this organism here, this organism actually anchors to the substrate, or the base below. This would usually be in a pond or an aquatic environment, so it'll anchor to the bottom of the pond, and then it doesn't really move, so it doesn't use the cilia for movement. It just stays in one place. But the filters on the top here act as a little mechanism to catch food particles, and food will actually stick to these hairs, and then they can actually be slowly drawn inside. So they act almost like a filter, literally, where they filter incoming food, they stick to those cilia, and then they can be kind of absorbed, almost like a spider's web, catching little particles of food. Okay, so that's the eukaryotic cilia there in a nutshell. Same structure, similar function, but then the extra filter feeding capacity there. Now, those are the common types of appendages for eukaryotes, and we'll just do one more little quick section here over the membrane, and then we will take another break for review. So eukaryotic cell membranes is the next topic, and if you remember back in chapter 4, I talked about membranes there as well. And in both types of cells, in all living organisms, a cell membrane serves the same function. And so let me just clarify, all living things will have a cell membrane. I mentioned that in the diagram showing the eukaryotes versus the prokaryotes. All living cells have these, and they're pretty consistent. Here's a quick diagram of what they look like. You have 2 layers of a molecule called a phospholipid, and these phospholipids shown here move their heads. They have 2 components, a head and a tail, and the heads line up on the outside. These little balls here represent the heads, and these little sticks here in the middle represent the tails, and they line up inside. And this creates a watertight region from the outside and inside of the cell. So this zone here in between is actually completely free of water. It's called a water-fearing or hydrophobic region, and it creates a distinct boundary where fluid tends to stay on one side of the cell or the other, and usually there's fluid on both sides typically. Now, things can move through here, but only through certain passages. So if you can think of this as a wall that then has gates and doors. So here's another figure, and this shows a protein pore, or the equivalent of a gate or channel, and these will often span the entire width of the membrane, and act as a key site for things to move in and out. Okay. Now we have lots of other things embedded within a membrane, and I'll mention a few of these, but certainly not all of them. But the idea is that, like the prokaryotes, we find this again in eukaryotes. It's consistent amongst all living things. That it's made up of a phospholipid bilayer that should be reviewed from general biology. I know not everybody's had it, but that's a major topic there. And ultimately, the membrane is said to be selectively permeable because only certain things get in and only certain things get out, hence the selective. And then permeable, meaning to permeate, move in and out of something. Another question on the review has to do with the proteins. So a question about what are the functions of proteins in the membrane. So to clarify, this membrane is made up of a lipid, a fat, the actual barrier here itself with the heads and the tails. The rest of it, like these protein pores here, they are variable in their function. So some examples of what they do, they act as transport. As I mentioned, things can move across here from one side to there, so transport across the membrane. Keep going the wrong way. In cells, these also act as an identity tag. Your cells each have their own specific marker, and for example, a protein like this can actually stick out of the membrane and on it carry a unique little molecule, much like the flag of a country, that is unique, in this case, to each cell. And it's like a fingerprint is probably a better way to think of it. It's like a fingerprint for a cell, and that's where the ID comes from. And they, unlike a fingerprint, they are just made up of different specific things that will characterize unique patterns, things like that. And communication, cells can send signals back and forth through proteins on the outside. I don't have a good diagram for that. That's a little more complicated. Think of nerve cells and how nerve cells interact together. And those proteins function to allow two cells to interact. And they also allow for attachment. Cells can attach to things with those proteins. Now, a feature that is unique to eukaryotes, and a feature that provides a greater amount of stability and strength to the membrane, are structures in really chemicals called sterols. Okay, so you want to recognize that term. And a sterol is really a type of molecule. Here's where they're showing cholesterol is an example of a sterol. So here's one showing the cholesterol molecule here, another one here, here, here. So you see they're kind of scattered throughout the membrane here. And sterols, like cholesterol, which is an example of a sterol, what they do is they strengthen the membrane. So the phospholipid membrane is relatively loose, and it's not very cohesive by itself. Now, in smaller organisms, this may function okay because their cell size doesn't ever get too big. But remember I mentioned as you get bigger, you need more infrastructure, more reinforcement. And one way that eukaryotic cells get this is by strengthening the membrane itself. And it does this through molecules called sterols. So just think of them as kind of a reinforcing agent for the membrane. It allows it to maintain some fluidness. It can still bend and stretch and give like a plastic, but like a plastic is strong at the same time. And that's important for certain cell types. So sterols provide strength and rigidity so they can remain flexible but strong. And an example is cholesterol. And so, cholesterol is actually a totally necessary molecule. It's often discussed in a negative, too much, overabundance context of human health. Usually you hear about cholesterol when it's bad and you have too much of it. But it's actually a fundamental molecule in all eukaryotic organisms. We all have some kind of, I should say sterols are, cholesterol is common to all animals, but all eukaryotes have some kind of strengthening agent in their membrane. This again allows them to get bigger and yet still remain somewhat strong and inflexible. Now, the next section here, eukaryotic cell walls. Eukaryotic cell walls are functionally almost identical to those of prokaryotes, and that is they help strengthen the cell and they help reinforce it. So this is another factor that can give each cell additional strength in structure. Now, one thing that we see in eukaryotes is that fundamentally speaking, some animals have them, or some cell types have them and some don't. Whereas in the prokaryotes, really we described most of them as having some kind of cell wall. We talked about the gram negatives and we talked about the gram positives. and the differences in the cell wall. But generally speaking, most of them have a cell wall. In the eukaryotes, it really depends on what group you're talking about. So, for example, the plants and the algae, if you remember, are actually a type of protista. I talked about those back in chapter 1. And then the fungi. Plants are one kingdom, fungi are another kingdom, algae are a group within the protista kingdom. All of these are more or less characterized as having cell walls. And each one will have different types and slightly different molecules that make them up, but they can more or less be characterized as containing cell walls. Plants and algae contain a cell wall made of cellulose, and fungi contain a cell wall made of a molecule called chitin, which is a different building material. In animals, however, animals being another kingdom, and then protozoa also being a member of the protista kingdom, along with the algae, they completely lack cell walls and never have them, and have additional cholesterol type or sterol type reinforcements in the cell wall, or the cell membrane, excuse me. So these are even more important in the animals in the protozoa that lack cell walls. Sterols are found in both, but in organisms that don't have the extra protection of a cell wall, sterols play an even bigger role and allows them to remain strong and yet flexible. Okay, so the cell wall, I really just want you to recognize what types of animals, what types of organisms, have them, what don't, and then connect this back here to the sterols being even more important to help strengthen that cell membrane. Okay, here's a figure showing the cell wall of a plant here, this dark green structure, and this is a cross-sectional view here of chitin, which is found in the fungi, showing a different type of cell wall here, reinforcing just above the membrane of fungi here. Okay, so we'll go ahead and stop, take a look at the part 2 review, and we will start the next section here. Okay, I mentioned the cytoskeleton earlier, and this is another topic that's tough to get into any detail because we could talk about it all semester. But in a general sense, I want you to understand what it is and how this is important. So the cytoskeleton consists of a series of structures that are found inside and then connecting towards the outside of the cell. And you can really think of these as a scaffolding for which the outer edges of the cell, like the membrane and the wall, can attach, and then as another anchoring mechanism inside the cell to reinforce all these structures here. So again, these cells get bigger and bigger and bigger, and if they don't reinforce themselves, they'll simply implode on themselves. So they need some kind of way to brace those structures to increase the size. And really, we see that the cytoskeleton provides just that. It provides that reinforcement to allow that larger and larger size to exist, and at the same time not implode. Now, a little bit more specifically, the cytoplasm can function in addition to anchoring the organelles. So inside, these organelles can actually be positioned and even moved using the cytoskeletal features as both an anchoring mechanism and then sometimes also as kind of like a pulley system to move them around the cell. So that connects to the next part here, moving molecules and vesicles around the cell. These can actually move things around. They can almost act like a little road-like structure within the cell, and little units can actually pull vesicles along them. And I can pull up a video here to show that in a minute to give you an idea what that looks like. And then also the whole cell can change its shape in conjunction with the movement of these structures here. And this allows for the cell to kind of invaginate or to move in one direction. Again, I'll pull up a few examples of that here in a second. Now, from a technical standpoint, the cytoskeleton is made up of at least 3 components, and those are listed here with a little bit about each one. So, actin filaments, microtubules, and then intermediate filaments. Now, I'm not going to get real technical and testing you over these. This is something that I just mentioned a little bit, so I just want you to recognize the 3 and then recognize a brief description of each. So actin filaments are often found right around the cell membrane. So these are closer to the periphery of the cell. And one of their biggest roles is for movement and contraction. And the way these work, tell you what, let me pull up a real quick figure here. Let me pause it real fast. Okay, let's take a look real quick. Actin filaments are made up of individual components, and these can be arranged one by one and I found a little quick video in a structure called actin polymerization. This is real short and shows individual units. These are the actin units, and they self-assemble or polymerize spontaneously. This is an animation, obviously, but intended to show what that would look like, just real quick. Okay, that's polymerization, self-assembly. The way these work within conjunction to movement is that they will form near a boundary and literally push the cell boundary forward. Now, this is right in the middle of another video that's talking about a specific type of organism, but it's going to show how the polymerization here would push a cell outward.
Cell pushes the lamellipod forward. This movement is driven by rapid polymerization of actin filaments at the leading edge of the cell membrane. In the next stage, the lamellipod adheres to the underlying surface.
Okay, and it goes on to talk about how that pushing forward ultimately moves the cell forward, kind of inches it along. Okay, so that's just a real quick visual on how actin moves a cell. Microtubules are what we talked about as the structures within the flagella. Just to go back and clarify, the 9+2 arrangement, these are actually made of microtubules. The 9+2, those are in reference to microtubules. So these are long, hollow tubes, and they have a variety of functions. And so we talked about how they use a flagella, or they're used in flagella and cilia. They can also move things inside the cell. Kind of like the actin, they can spontaneously form and move structures from one side of the cell to another. So usually moved or used to move things inside the cell as opposed to moving the cell itself. The last type are called intermediate filaments, and the name intermediate implies that they're found within the cell. And these often act for structural reinforcement of the cell, maybe to clarify reinforcement internally, which then helps reinforce the internal cell organelles. So if we go back here just real quick, this is the figure out of the text. It's showing actin filaments near the surface. Microtubules can be found usually internally as well, or along with the intermediate filaments, excuse me. So actin filaments on the outside, intermediate and microtubule filaments on the inside. Intermediate filaments playing more of a structural role. I know this figure doesn't show that all that well, but that's the idea there. Okay, so recognize the three basic types and the three basic functions of each there. Last little bit, we'll summarize the eukaryotic organelles, and then we'll go through the specifics of each next. So the last of this part, I should say. So to clarify, the next topic, we're going to get into the organelles in a little more detail. And before we do that, we just want to mention what the function in general of organelles are. And they're essentially isolated locations that provide a unique function in each spot. And as again, here's my analogy. If I haven't beat that to death yet, I will eventually. Different rooms of a house, different functions within each room, okay? And each organelle then acts as its own unit with its own function. Now, adding to this, organelles are partly defined by the fact that they have their own membrane. So they literally have their own walls, not literally, but figuratively, I should say, they have their own walls within the cell. And that's made up of the cell membrane-like structure, the phospholipids. To go back just real quickly, it's very similar to this type of structure here. So within the cell, going back here real quick, looking at this cell, pull this figure here. Each one of these structures here has their own little membrane made of the phospholipid-like content that separates it from the rest of the cell. So the cell here has its own membrane, and then structures here also have their own membrane. So membranes within membranes. And that's one of the things that partly defines what an organelle is here. Okay, but ultimately they're isolated locations with their own membrane and their own functions. And again, collectively, they serve to make each part of the cell dynamic and function separate from the rest of the cell. Okay, go ahead and pause, take a look at part 3, and we'll go to the next part. Looking at part 4, we're going to talk about some of these specific organelles, and I'll do this really in 2 parts here. So in the first part, we'll look at the nucleus. And the nucleus is what I mentioned earlier as one of the key primary differences between eukaryotes and prokaryotes. So keep that in mind. Now, here's just the isolated section of the nucleus. And what we're really looking at here is this envelope what's called the nuclear envelope, and this is again a separate membrane that isolates this portion of the cell from the rest. So the nuclear envelope is the membrane portion that physically surrounds the nucleus. Now, in addition, the nucleus will be studded with what are called nuclear pores, and these are little openings, little passages that allow transport in and out of the nucleus. So it's not isolated, but it's separate, but still connected in that these pores allow entry and exit of the necessary features that go in and out of the cell. And the last part is called a nucleolus, and this is kind of a production center inside the nucleus. It's where a lot of, or not a lot, but certain key things are actually produced and then often exported outside the nucleus. Now, that may not make a lot of sense just yet, but the idea is that the nucleus, again, is its own organelle for the DNA, and we're looking at the components here that make up the nucleus itself. The envelope, the pores, and then the nucleolus, a production site within the nucleus. Now, what's not shown here is all the DNA, to clarify. The DNA would more or less just fill this to capacity, and it would be completely saturated. So they don't draw that. I guess they don't animate that for that reason. But this would actually be filled with DNA. Otherwise, it's being shown as empty right now. Now, inside here, the DNA would actually be in a form called chromatin, sometimes pronounced chromatin, or what are more familiar as chromosomes. Let me pull up a figure real quick for that. Here's a figure showing a nucleus. Looks like a bowl of noodles, but this is more accurately showing what it would look like full of the DNA. Now, in a typical form, it's in what's called chromatin, or I should say under typical conditions, it's in a form called chromatin. And chromatin is really a loose kind of spaghetti thread-like form that's actually wrapped around little proteins called histone proteins. So this DNA can stretch for really long distances, but it's ultimately connected around these little wrapping proteins that bundle it together. Now, these bundle and they overlap and wrap again back on top of each other, and in certain conditions such as cell division, when a cell is getting ready to divide, all this DNA will be wound up into what we call a chromosome, a condensed chromosome. So a chromosome is really a big long piece of thread-like DNA wrapped into a nice, neat, organized structure that often looks like an X here. So that's a little bit about the actual DNA itself. Chromatin is the loose form. Chromosomes are the condensed, wrapped form that would occur prior to cell division. So another organelle common to all eukaryotes is an organelle called the endoplasmic reticulum, and this will essentially hang off of the eukaryotic cell's nucleus. So here's, going back to this figure, here's the nucleus here, and endoplasmic reticulum is shown as this other purple stuff here that is loosely attached to the nucleus. So the nucleus is just this structure right here, and all this stuff hanging off of it, shown here, here, and here, all in purple. This is all part of what's called the endoplasmic reticulum. And the endoplasmic reticulum is a more advanced way of routing things in and out of the nucleus. So if you think of the nucleus as kind of the brain of the cell, the endoplasmic reticulum is essentially the direct passageway in and out of the brain, and things can be sent directly from the brain to the rest of the cell through the endoplasmic reticulum, and then other things can enter the cell through the endoplasmic reticulum. Now, sometimes they can enter via other ways, but this is kind of a key center in and out of the cell, or in and out of the nucleus of the cell. So a little bit more specifically, you have two types of endoplasmic reticulum, and each has a more specific function. So, for example, one of the types is called rough endoplasmic reticulum, and this is abbreviated as RER. It's considered rough due to the fact that it contains structures called ribosomes, and those are the protein synthesizers we mentioned. If you look here, these little blue dots, these are the studded ribosomes, giving it the quote-unquote rough appearance. And wherever these are found, the endoplasmic reticulum is considered rough. And in this scenario, the presence of the ribosomes actually dictates the function, which is protein synthesis. So really what the rough endoplasmic reticulum is a special portion of the cell where ribosomes congregate, giving it the rough appearance, And ultimately, this whole area specializes in protein synthesis, but then also it functions in transporting the proteins, both in and out of the nucleus. So just recognize that there's two types. One is called rough, and it's rough because of ribosomes, and ribosomes synthesize protein. And therefore, this whole structure, the rough endoplasmic reticulum, functions and specializes in protein synthesis, which is important for the cell. Proteins are pretty much important for all living things, so this is a really kind of central area where this important process takes place. The other type is called a smooth endoplasmic reticulum, and it's considered smooth because it lacks ribosomes. So you have a rough with and a smooth without ribosomes. Now, where the ribosomes don't exist, the function actually changes a little bit. And in the smooth endoplasmic sections, or the SCR segments of the endoplasmic reticulum, what we see is that functions tend to shift towards more general things such as nutrient processing. And another thing that I forgot to add to this, I need to go ahead and clarify that. Another key thing that they do, the SCR does, I should say. Let me just go ahead and clarify here. Whoopsie, I started. Sorry. Oh, okay. Apparently, Shift S starts my camera. Okay, SCR, smooth endoplasmic reticulum, functions primarily include nutrient processing. And another big one, which I didn't have listed there, which is why I'm putting this up here, is actually detoxification. Detoxification meaning it breaks down harmful substances. So, example given, the breakdown of alcohol, which by all means is a toxin. So when you put alcohol in your body, the liver, most of us kind of know that the liver is what processes alcohol. But even more specifically, it's really the smooth endoplasmic reticulum within the liver cells that are specialized at doing that. And the truth is, all of our cells can break down toxins. Some are just better at it than others. The liver is specialized at that. But the idea is every cell has kind of its own detox center, and the SCR does just that. Now, there's lots of other things there, but keeping this somewhat simple, we'll keep it to that. So, nutrient processing, that, by the way, that means a lot of different things. We're really generalizing when we say that there. That doesn't really mean much to you, so I apologize for that. But, again, we have to stay vague to keep this somewhat manageable here. Focus, just understand it processes nutrients. You know, if you want to know more about that, I'd be happy to explain it to you. But the other thing there is then it detoxes. So, that's a kind of a very particular feature of this part of the cell, the smooth endoplasmic reticulum. So these are two functions here connected to the nucleus. This focusing here mostly on the rough section, and here you can see those ribosomes, which is what they would look like if you got real close to it there. Okay, the next organelle is called the Golgi apparatus. And the Golgi apparatus is a real interesting one, very mysterious in my opinion. Now, to start describing it, we'll use an analogy that's been given. This is not my analogy. This is one that's been passed down for a long time. And the analogy is that it's often referred to as the UPS, quote-unquote, or the post office of the cell. Or you could say even the FedEx of the cell. The idea is that like UPS, like the post office, like FedEx, the Golgi apparatus receives packages from other parts of the cell. It actually can label those packages and then reroute them to different areas of the cell. So it essentially receives a package, labels, and then reroutes that package to a new part of the cell. And that's why it's often referred to as the UPS or the post office, because just like the post office, they receive a package, they put their own label on it, and then they send it somewhere, right? Now, the difference between the Golgi apparatus and the US UPS or the post office is that the Golgi apparatus will modify that package along the way. And theoretically, UPS or post office should not modify your package, only reroute it. or distribute it. But it's very, very close analogy there. The difference is that the Golgi apparatus kind of makes its changes along the way. But that's essentially what it's doing. So here's a real quick figure out of the book. And the way this works is that the endoplasmic reticulum, both the rough and smooth parts, will synthesize products. We mentioned that the SER processes nutrients. So some of the processed nutrients, for example, will actually exit the smooth endoplasmic reticulum in a little structure called a vesicle. And this is part of the membrane that just pinches off from the rest of the cell. Now it will actually travel across the cell, usually attached to microtubules, not shown here. And those microtubules then will allow that vesicle to move to a different part of the cell where it meets the Golgi apparatus. So on one, they show it right next to each other. There's actually some distance in here. But as it moves from one side of the cell, from the endoplasmic reticulum to the Golgi apparatus, it will actually fuse. The contents of this will fuse, and the what's inside will dump into the Golgi apparatus here. Now, as it dumps inside, it will move through these different layers from one side to the next. And ultimately will come out the other side here. And when it comes out the other side, it's been relabeled and redirected to go to a new location in the cell. Let me pause real quick and find a real fast video to visualize that for you. Okay, here's a real quick video. This one's perfect, real short and sweet. And what's happening, here's the Golgi apparatus. And what it's going to show next is it's going to show vesicles from the endoplasmic reticulum branching off, fusing, moving through the Golgi apparatus, being changed, relabeled. They come out the other side in a new package. And then what it's not showing there is that package will then go on to be rerouted somewhere. These packages can sometimes stay inside the cell or exit the cell and be dumped externally for some purpose outside the cell. So the Golgi apparatus is very mysterious in that it seems to do all this autonomously. Now there's a complex set of mechanisms behind the scenes here that explain how it works, but that's kind of a cool thing. It's taking products from one part of the cell and relabeling them, repackaging them, altering them a little bit, and then ultimately sending them where they want to go. A real quick example of an example that might pertain here. There are enzymes created inside the cell that would need to function outside the cell. So in some cases, the Golgi apparatus would actually take these to the cell surface and dump them. So here's a real quick image of what that might look like. Once again, you see packages from the ER, the endoplasmic reticulum, coming to the Golgi apparatus, being processed as they move through these layers, coming out the other side. And now some of these packages may ultimately then move from the Golgi apparatus to the cell membrane, where they would fuse and then be dumped externally. So enzymes, for example, that would digest food outside of the cell would be excreted in this kind of way. This is also how your brain functions 24/7, 365. It's constantly creating neurotransmitters like dopamine, like serotonin, like pseudoephedrine. This is a real quick figure of the Golgi apparatus showing how things might be secreted. Here we see the Golgi apparatus and products from the endoplasmic reticulum being sent over, pushed through the Golgi apparatus, and then ultimately coming out the other side. And in some cases, what would happen is those products from the Golgi apparatus, the packages being sent, would actually fuse with the membrane and then be dumped outside the cell. So you'd have a externalization of those products. They didn't literally be sent outside the membrane. So that's one way those work. That would take place with, for example, digestive enzymes that would need to do their effect outside of the cell. Also, your brain cells function this way. Neurotransmitters such as dopamine and serotonin are often secreted outside the cell to act on other cells in this kind of way. So that's the Golgi apparatus. Okay, go ahead and stop here at part 4. And we've got one more part to do with the last little bit remaining here. Okay, the last part, starting with the mitochondria, looking at a few more organelles here. Mitochondria is unique in a lot of ways, but we'll focus on the basics. And what you want to know about it is that it is a organelle that provides cellular energy, and it's considered the quote-unquote powerhouse of the cell. Now, in general biology, a topic that you should have learned and is pretty central to that class is called cell respiration, the process by which glucose is converted to energy. And in eukaryotes, this takes place mostly in the mitochondria, almost exclusively here in the mitochondria. So this is like a little center within the cell for energy. Think of it like the gas stations of a town. This is the special point where energy for the cell is created. Now, on a side note, these are actually thought to be ancient bacteria trapped within eukaryotic cells. If you look up, type in Google endosymbiotic theory, you see there's tons of evidence, and more or less this is accepted scientific. There's an accepted scientific consensus that these are recognized as being an ancient bacteria that was symbiotically trapped within eukaryotic cells, and ultimately became kind of a slave to the larger cell in that it functioned to produce energy for that cell. Now it also gets its own benefits from being there, but essentially it's a living cell within a cell. Now today they've become more or less just a functional part of the cell, but the idea is that these were once thought to be a living organism separate from the cell. But ultimately we don't want to get into that too much. That's a separate topic if you want to look that up. But for the most part, sticking to the basics, it's another organelle, and its function is for energy production. Okay, I'm trying to keep this short here, so I'm trying to wrap this up, and I apologize if I'm going too fast. But the next organelle are called chloroplasts. Chloroplasts, like the mitochondria, are also thought to be another symbiotically trapped organism. And like the mitochondria, they also function in energy production. Now, chloroplasts are specific to photosynthetic organisms. So chloroplasts essentially are what conduct photosynthesis. So when we talk about a photosynthetic organism, remember that the examples are plants and algae, and then there's some bacteria, but this would not apply to those bacteria. So plants and algae, the major photosynthesizers, they actually get that property of photosynthesis from the chloroplast. And this is another special little center for energy. Now, the chloroplasts produce the energy, and then the mitochondria break that energy down. So there are really two different aspects of energy generation. Now, plant cells will have both, to clarify. Plant cells will have chloroplast and mitochondria. And that means they can both produce their energy in-house, so to speak, and process that energy at the same time. So plant cells do both. They make their own energy and process it internally. So they have both types. Animal cells don't have a chloroplast. They will only have mitochondria. And this is also true of protozoa and fungi. They don't have these energy processors. So these would only be found in plants and algae. And there's another side note here. These are the major generators of organic matter on Earth. These little structures here produce the vast majority of all living matter that you see, from tree matter to pond scum to the tissue of all living things. Ultimately, whether it's directly or indirectly, is generated through chloroplasts. So they are really what's responsible for everything on Earth, for more or less the organic matter anyway, being here. So that's kind of an interesting side note there. So they're pretty significant. The last part are the ribosomes. We mentioned ribosomes in Chapter 4. And just like in Chapter 4, I want you to continue to recognize that they are protein synthesizers. And that's kind of the general function that you want to recognize. In a little more detail, they consist of two subunits. And these subunits sandwich together to create the ribosome as a whole. So the ribosome consists of what's called just referred to as a large subunit and a small subunit based on size. And they essentially sandwich together and collectively make up the ribosome. Now these two units can vary. The exact shape and size can look different. And what we see is that while all organisms have them, there are two general types that relate to whether an organism is eukaryotic or prokaryotic. So you have what's called a eukaryotic ribosome, which is referred to as an 80S ribosome, and then a prokaryotic ribosome, which is referred to as a 70S ribosome. Now, where that naming comes from, I don't want to get into. It has to do with the way they test them for size. But essentially, you've got a large and a small group of or series of ribosomes. So while they do the same thing, they are structurally different, and ultimately one's a little bigger and a little bit more complex than the other one. And go figure, it's the eukaryotic ribosomes that are bigger and more complex. Now, this does have, by the way, here's a figure showing a ribosome from a 3D animation, and this is RNA being processed through here. I mentioned that way back in chapter 4. Chapter 4, I drew a little figure of RNA moving through a ribosome, and so this is kind of what that would look like here. And we'll come back to what's really happening with this later. But the last thing here I want to leave you with is a significant an example of why the difference between an 80S and a 70S ribosome is significant. Now, generally speaking, they're different, and there's lots of subtleties there. But a big picture thing that's significant, and this would apply in other contexts, but you have a mechanism by which certain drugs work, and that mechanism is called selective toxicity. And what that means is that a drug will target a feature that's unique to one cell and not found in another. So if we only want to kill bacteria, for example, that we have in our body during an infection, one way to do that is to target a feature that is unique to the bacteria that's not found in our cells as eukaryotic organisms. So as humans, we're eukaryotic, and the bacteria that may be infecting us, they're prokaryotic. So if we can find a difference fundamentally between these two cell types, we can target that difference that occurs in the prokaryotes and theoretically have what's referred to as selective toxicity, a drug that only targets a microbe like a bacteria and not a eukaryotic cell. So this is something that can apply in reference to the ribosomes. So there are drugs that specifically target the 70S ribosome and not the 80S ribosome. And because the 70S is only found in bacteria, that drug becomes selectively toxic to bacteria and not the eukaryotic organisms, people and animals that are host to these bacteria. So some examples would be drugs like tetracycline. Tetracycline has an active ingredient that targets the bacterial ribosome. There's actually a lot of them. Many antibiotics function in a way kind of like this. But if you want to look one up here, I'll type that out for you real quick. You type in tetracycline. You don't have to do this, by the way, in case you're interested. Tetracycline is one. Doxycycline is maybe one you've heard of. Doxycycline is a type of tetracycline. Or erythromycin is another one that's functions like this. Chloramphenicol. These are all examples of antibiotics that target ribosomes, and as a result, are selectively toxic to bacteria. Now, this applies in a lot of other contexts. All drugs are somewhat selectively toxic. This is just one example of how that would apply in a way that makes a drug specific to bacteria. So to clarify, you target the ribosome type found in bacteria, which is different from the ribosome type found in the eukaryotic host, and as a result, the drug theoretically hones in on just that structure. Now, when the drug targets the ribosome, to finish this here, if the ribosome is disabled by the drug, the cell dies. Without ribosomes and without the ability to produce protein, all cells will die within a short amount of time. So by disabling the ribosome, you essentially kill the cell indirectly. So that's the idea of selective toxicity and how that would apply in an example where a drug might target the 70S and not the 80S. Hopefully that makes sense. If not, I'd be more than happy to go over that further in lecture. I realize I'm going a little bit faster than I'd like to, but as promised, I'm trying to keep these to under an hour and 15 minutes. So I'm right there. I'm going to edit a few minutes out of this, and it'll be a little bit less than an hour and 10. So get back with me with any questions, and I will see you next week in our face-to-face lecture. And I will leave you here with the Part 5 review questions. All right, have a great day. Bye-bye.